guys, so here we go. This is going to be on mitosis, and it's a little cold in my office today, and I'm trying to sit up tall so I'm not cutting myself off. Okay, so cell division, growth and repair, tumors and cancer. And so basically we grow by reproducing our cells, creating more cells. So what you think of when you think of a cut, that's your repair. So like when you skin your knee and you form a scab to block up the blood until the new skin cells have time to divide, increasing their numbers to patch up the hole here. Then we have when the egg and the sperm unite, then we start off as a single-celled organism called a zygote. And then that will then, through cell division, or the process known as mitosis, creating identical cells from the original parent cell, you will end up with a cute little baby. So it goes from 1 cell to 2 cells to 4 cells to 8 cells eventually, increasing its number, doubling each time until eventually differentiating, becoming, muscle, digestive, all that stuff. Now, sometimes, why is this important? Again, because when things don't happen properly, when mitosis isn't regulated properly by the cell, then you can get something like you see here, these web toes, or what you see here in a lung. You can see this big, massive tumor right here, and some more tumor down here and tumor down there. Again, that's when mitosis isn't functioning properly. It's the failure of mitosis to stop, which leads to tumor formation. Okay, so we're talking about cell division, mitosis, increasing the number of our body cells, also known as somatic body cells. This is again for growth and repair. We call it mitosis. It is the division of the nucleus, After the contents of the nucleus, which is our genetic material, which controls the cell, is divided, then the cell will go ahead and divide the rest of the cellular contents through the process of cytokinesis, which is division of the cytoplasm. Now, every cell doesn't live forever. They do have to go through a programmed cell death, and this is called apostosis, or programmed cell death. So when cells lose their ability to regulate apostosis, that's when they become precancerous, tumor forming, and then can become cancerous cells. Now, every cell goes through a cell cycle, a cell life cycle. So if we start right here, this is like one of your skin cells just divided, and it's going into its life cycle. So this whole part, G1, DNA replication, S phase, and G2, we call that, so this blue range right here, that's called interphase. It's the time between cell divisions, and that's when the cell is doing its normal job functions. For example, if it's a skin cell, it's doing its skin cell jobs. If it's a muscle cell, it's doing its muscle cell jobs. And then this is subdivided into G1, which you can see growth and normal metabolic roles. S phase, which is DNA replication. This is when the cell is going to make an identical copy of its genetic material, all 46 DNA strands. Remember, they're not chromosomes yet. Then G2, growth 2, preparing for mitosis. Then we have the mitotic phase, subdivided by prophase, metaphase, anaphase, and telophase. This is when the nuclear contents divide, and then cytokinesis will be right. It overlaps a little bit over telophase. Telophase and cytokinesis overlap a little bit, and then you'll have 2 new cells. Basically what I just said here, you've got your interphase, the time between cell divisions, your G1, the cell is growing and normal functionings. S phase, this is when the DNA replicates itself. So again, like this little cartoon here from Thank You Amoeba Sisters. And so notice here that on the copy machine, it's a single-stranded chromosome, and then when it comes off, then it's a double-stranded, okay, because it's made an identical copy of itself. And the centromere, holding the 2, now we call them sister chromatids, okay, chromatids. G2, the cell prepares to go into division. And then G0 is the halted phase. Not all of our cells continue to divide throughout our lifetime. Some of them enter a G0, a halted phase, example being nerve and muscle tissue. That's why if you get nerve damage, it's permanent because those don't repair themselves. And then, of course, we have the mitotic nuclear division and cytokinesis. The chromosomes are coming. The chromosomes are coming. I really like this here. Prophase, okay? So we don't visualize chromosomes because they're not condensed and formed until prophase, which is the beginning of mitosis. This is when the DNA will condense into those classic chromosome shapes. Now, the chromosomes, since the DNA is already duplicated in S phase, when it condenses, it is what we think of as the classic X formation. So what's going on in prophase? The phases of mitosis, you can kind of think of PMAT, prophase, metaphase, anaphase, and telophase. So most of the activity is happening in prophase. It's the longest phase. So what happens, and here's an actual cell, okay, going through a picture of one going through prophase. So what happens is the DNA is going to condense, and by the end of prophase or late prophase, you will see the individual chromosomes, double-stranded chromosomes. What else is going to happen here? The nuclear envelope is going to disintegrate, okay? It disintegrates. And so does the nucleolus, okay? The nucleolus inside the nucleus. The spindle fibers are going to start to appear. They're like marionette strings in between these two organelles called the centromere. Not the centromeres, the centrioles. The centrioles. So the centrioles, what they do is they start to move towards opposite poles, and the spindle fibers form between them. So by the end of prophase, what you have is the centrioles are at opposite ends of the cell. The spindle fibers are attached to each chromosome at the centromere. So that's what you have there in prophase. Next, metaphase. So you can think of the middle, okay, the middle. So the spindle fibers, they're going to contract their protein fibers, and they're going to contract and move those chromosomes into the middle of the cell. That's metaphase. Then we have anaphase. So you can kind of think of moving apart. So what's going to happen is each centromere, okay, and here's an actual picture. Here, each centromere splits, and that's going to separate the two sister chromatids. The spindle fibers contract, separating the sister chromatids from each other, moving to opposite poles of the cell. Then we have telophase. Okay, so in telophase, this is when the chromosomes have reached the opposite ends of the cell, and you can see here that cytokinesis is starting to overlap here as well. You can see it's pinching in right here. So the now single-stranded chromosomes, because they were split from their sister in anaphase, they stop moving and they're going to unravel. So the chromosome is going to, what we think of as, disappear, okay? Because again, it unravels into single-stranded DNA, so you just can't see them anymore. The nuclear envelope will then reform around each new nucleus, and the nucleolus will reform, and then the spindle fibers disintegrate, and the cell will split in half, and then you have two identical diploid daughter cells. Okay? So, like this, stop staring at my cleavage, because the point where the cytoplasm does pinch in half is called the cleavage point. So, again, the cell pinches in half, separating, so you get two new identical diploid daughter cells. Okay? Alrighty, so there's a connection between mitosis and cancer, and so I will post this link separately. I will post this link separately, and you can see that it is going to be a link to the, I have it here someplace, should be to the Amoeba Sisters, okay, cells and cancer. I do want to show, where did you go? Blood Lily, there you go. I did want to show this short video here of mitosis time-lapse photography going on in a blood lily. So you can see the chromosomes have formed, so that was prophase. Now you've got metaphase happening where the chromosomes, oh, there's anaphase, okay, they were moved to the middle. Now they're separated, and now we're in telophase. And notice how it looks like the chromosomes are disappearing. Do it again. And so here again we've got prophase, late prophase, because the chromosomes have formed. Metaphase, they're moving to the middle of the cell. Okay, moving to the middle of the cell. They're going to split. When you see them start to split, that's anaphase. Okay, so they're moving, 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 moving. Middle of the cell, middle of the cell. Here they go. They're in the middle of the cell. All right, there you've got your anaphase. Okay, and I'm going to pause this real quick. Ooh. So see over here and over here how it looks like chromosomes are disappearing. That's because they're not disappearing, they're unraveling. Okay, they're unraveling back into the really thin strands of DNA that you can't see. Okay. Another thing I want to point out here is that in plant cells, unlike animal cells will form that cleavage and pinch in half. But what happens with plant cells, because they have the cell wall, it can't pinch in half. So basically what they do is they build a wall between them. Okay, so yes, they will build a cell wall in between in plant cytokinesis, therefore separating the two cytoplasms of the two newly formed identical diploid plant cells. Great.